All right, so just a quick recap of long bones. We went over this in class. Long bones, some of the basic parts. You have the diaphysis, basically, that describes the long shaft of bone. It's a collar of compact bone that surrounds the medullary cavity, filled with a yellow marrow, which is basically fat storage. Its function is for rigidity. You have the epiphyses or the epiphyses. Those are the expanded ends of the long bone. It's a spongy bone filled with red bone marrow. And because it's filled with that red bone marrow, you do have that hematopoietic tissue in there. It forms synovial joints with other long bones. Then you have the periosteum, and basically that is going to be described as that area of the bone where you're going to have that dense, fibrous connective tissue that is going to surround that outer surface of bone. You also have an inner layer of osteogenic tissues that is composed of both osteoblast and osteoclast cells. You also, in the periosteum, have the nutrient foramen, and that is going to serve as the passageway for nutrient arteries to penetrate bone. Overall, the periosteum is provided for protection, and it's an attachment site for muscles, and it also plays a role in bone remodeling. The endosteum, on the other hand, basically that is just the inner lining of that medullary cavity with an osteogenic layer as well. So the osteogenic layer, you have both osteoblast and osteoclast. And remember, those are bone cells that are going to build bone and cells that will break down bone. And the endosteum is lining and also plays a role in bone remodeling. At the end of the bone, you have articular cartilage that's going to cover the epiphysis, and that articular cartilage is basically going to act as a shock absorber and form a synovial joint. We'll learn about joints in the next chapter. The nutrient foramen, or the perforating canal, same thing. Basically, that is a perforated canal that is going to allow for blood vessels to enter and leave bone, and it's going to serve as nourishment. And then you have that epiphyseal line, and the epiphyseal line at a junction of the epiphysis and diaphysis is the remnant of the epiphyseal growth plate. And that epiphyseal growth plate we'll look at a little bit later. We'll talk about bone growth when we do after we do microscopic structure of bone. Long bones are different from flat bones. Remember that flat bones themselves are covered by a periosteum, which is covered by compact bone, and it's surrounded by an endosteum covered by spongy bone. Except in flat bone, the arrangement looks like a sandwich. So you have spongy bone that's going to serve as the meat sandwich between two layers of compact bone, which would be like the bread. And also in there, you do have the hematopoietic tissue that located in spongy bone is within the epiphyses of long bones and those flat bones. And remember, the hematopoietic tissue is going to be that red marrow. So overall, we do have the microscopic structure of bone. We do know that mature bone cells are called osteocytes. So where does this all come about? I think it's covered in on pages 7.4 or 205 and 206. You could see pages 7.4, figure 7.4 and figure 7.7 go over osteocytes. But overall, you have an organic component of bone, and you have this inorganic component of bone. And the organic component of bone, which makes up 35% of bone, includes cells and osteoids. And then you have the inorganic component, which are the minerals and organic compounds and ions that are going to come together to form the bone. So basically what we see is when we look at the microscopic structure of bone, we have those, the cells. These are going to be osteoprogenitor cells, and osteoprogenitor cells, which are the cells to which bone cells are going to come about, is derived from mesenchyme. And basically, osteoprogenitor cells can undergo mitosis and ultimately form osteoblasts, which are the bone-forming cells. Osteoblasts themselves are going to form the bone matrix by secreting collagen, and osteoblasts cannot divide via mitosis. So the osteoblasts are going to form bone matrix secreting collagen, and the mature bone cells are called osteocytes. So osteocytes are the mature bone cells that are basically come about from osteoblast, and it's the principal bone cell. Osteocytes cannot undergo mitosis, so they do not divide, and basically they are going to maintain that daily cellular activity, that is exchange of nutrients and waste with the blood. The extracellular matrix of the bone is largely collagen and inorganic salts. Collagen itself is going to give bone its resilience, and then the inorganic salts, like hydroxyapatite, is going to help make the bone hard. You also have osteoclast. We'll come back to that. You also have osteoclast, and basically that's going to function in bone reabsorption. So osteoclasts destroy bone or destroy that bone matrix, and that's important in developmental growth and maintenance, and also repair of our bones. If we look at the osteoid, primarily collagen, 90% of bone protein, which gives bone its high tensile strength. Other bone proteins include osteocalcin, osteonectin, and osteopontin. It also contains glycolipids and glycoproteins. The other thing with bone, the inorganic component of bones, which is 65% of bone, is that hydroxyapatite. Hydroxyapatite, if I come in here and spell it for you, let's go out here. So the inorganic component of bone is 65%, and what we do see there is hydroxyapatite. And basically, hydroxyapatite is the mineral salts that are going to form. Hydroxyapatite is composed of both calcium phosphate, and that's going to help give bone its rigidity. When you look at calcium phosphate, it's Ca3PO42OH2. All right, so looking at compact bone itself, we did look at this under the microscope. Basically what we have is that compact bone is that solid, dense, and smooth bone. The structural unit of compact bone is a thing called the osteon, or what we refer to as the Haversian system. In the middle there, we do have elongated cylinders that are cemented together to form the long axis of bone. The components of the Haversian system, or what we say is an osteon, are osteocytes themselves. And the osteocytes have a spider-shaped bone cells that lie in the lacunae, and the lacunae is what is going to be laid down within this matrix, or the lacunae has this matrix of collagen and calcium salts that is going to form this concentric layers called the lamellae. So these layers are basically going to be in concentric formation around this central canal called the Haversian canal, which you could all see it right here in this one osteon. And basically that central canal is where you're going to find both the blood vessels and nervous tissue. So in addition to that, you also have communicating canals within the compact bone. And communicating canals within the compact bone are things that we call canaliculi. And you can see that word right here. The canaliculi are going to connect the lacunae of the osteocytes. And then you also have these perforating canals, or what we call Volkmann's canals. And the Volkmann's canals are going to connect the blood and the nerve supply of adjacent osteons together. So we could see that all within this little area here. So what we do see is overall, when you look at those perforating canals, they are going to run at a right angle. So you can see that they run at right angles to and connect those adjacent central Haversian canal systems. So there you could see the different things of the compact bone, the perforating canals, the interstitial lamellae, osteons, and the central canals. Here it is at a higher magnification. Here you have the lamellae. Here you can see those communicating canals called the caniculi that are going to connect the lacunae of osteocytes. Here are the lacunae themselves. And basically there you could see some osteocytes, and there's that central canal. So this one whole structure is one osteon. In spongy bone, the microscopic structure of spongy bone, or what we also know as cancellous bone, basically that consists of this poorly organized needle-like pieces of bone called trabeculae. The trabeculae have a lot of open spaces between them, and those are going to be nourished by diffusion from the nearby Haversian canal systems. Remember that in diffusion, you have substances that are going to move from areas of high concentration into areas of lower concentration. The osteocytes themselves do reside in lacunae that are found within the trabeculae of that spongy bone. So if you look, here is the histology of spongy bone or cancellous bone. This is what it looked like. Here you can see the trabeculae. Here's the periosteum. And then down in here you have the marrow itself. And there is a really nice electron micrograph image of what the trabeculae, or those needle-like structures, look like within spongy bone. All right. I think we'll end there for the microscopic structure of bone. Our next lecture, we'll look at the bone growth and development. There is a pretty much a lot of detail with the bone development and growth that we will look at for both growth along the epiphyseal plate. We'll talk about the zones. We'll look at appositional growth. We'll look at also, as far as bone development, the homeostasis of bone tissue, and then factors affecting bone growth and development, such as nutrition, exposure to sunlight, hormone secretions, physical exercise. And then from there, we'll get into basic bone function and then briefly touch upon in lecture the basics of the skeletal system as we begin to dissect it down and learn all the different things. Thank you very much and have a nice day.